Welcome to solverfy.
So let's look at the overall process of how DNA can be extracted from cells. So you start with a few cells that you want to grab DNA out of. Say they are cancer cells and you want to sequence the DNA, or bacteria and you want to take out a plasmid so that you can insert DNA into that plasmid. You basically take the cells and you lyse them with lysing buffer and usually also centrifugation. And lysing basically breaks the DNA open, breaks the cells open. So breaks cell open so that the DNA is now out into the solution. And then once the DNA is out into the solution, you extract the chromatin. So chromatin is the DNA bound around histone proteins. Okay, so there are proteins that help the DNA get organized in the nucleus. The DNA complex with all the helping proteins is chromatin. So now you need to extract the chromatin out of that solution with extraction buffer, and then that buffer is something that can bind to the chromatin. So once you have that chromatin isolated, it usually precipitates to the bottom, and then usually you can take all of this liquid out on top and you are left with your DNA at the bottom. And then once you have the DNA isolated, you can either then sequence it or use it for DNA let's say GMOs, because genetically modifying organisms, because you need to recombine the DNA. That's what basically GMOs are organisms that have recombinant DNA. So you can make GMOs with that afterwards. So those are two options of what you can do afterwards. So let's look at our answer choices. What is the first step in isolating DNA? Well, first of all, before we even look at the answer choices, in the diagram that I've shown here, the first step is the lysing of the cells. Okay, this is cell number one. You need to break the cells open so that you can get the DNA out. Okay, so that's step number one, and that's choice D for us. Okay, lysing the cells in the sample. Let's look at why the other ones are wrong. Generating choice A, generating genomic DNA fragments with restriction endonucleases. That would be down here in the creation of GMO. So that's way after lysing. Okay, so that is after isolation of the DNA. So choice A is wrong. Choice D is wrong for the same. I'm sorry, choice B. Choice B is wrong for the same reason as choice A, because you are introducing the recombinant DNA into the GMO afterwards. And choice C is overexpressing proteins in E. coli. Getting DNA out of an organism or out of a cell has nothing to do with overexpressing proteins in E. coli. If you are isolating DNA from a cancer cell, what does E. coli have anything to do with that? So choice C is also wrong. So choice D is the only correct choice.